Hello there and welcome back. This is our third video in our video series for Chapter 6, Expression of Biological Information. My name is Miss Delia, and in this video we'll be going into 6.3, where we will look at protein synthesis, transcription, and translation. If you're ready, let's get started. So these are the learning outcomes for this subtopic. I hope that by the end of this lesson, you should be able to explain briefly what is transcription and translation, introduce the codon and its relationship with sequence of amino acid using genetic code table, explain transcription and the stages that are involved in the formation of mRNA strand from 5' to 3', and last but not least, explain translation and the stages that are involved in translation. So let's get started with protein synthesis. Protein synthesis is quite important because this is the process that produces proteins, and these proteins are things such as hormones and enzymes, which play a huge role in our body functions. Protein synthesis itself involves two stages. So we have the transcription stage and the translation stage. Eukaryotic cells, like yours and mine, have pre-mRNA process or RNA processing, whereas prokaryotic cells have no RNA processing. Let's talk a bit more about transcription. Transcription is a process in which genetic information in DNA is transcribed or copied to form mRNA, whereas translation is the process of interpreting or translating information, genetic information, or codons in the mRNA into sequences of amino acids or polypeptides. Now we talked about genetic code. So when we say genetic code, what do we mean? Genetic code is the set of rules that maps triple nucleotide sequences in DNA or mRNA codons to specific amino acids in the protein product. Genetic code is basically how to read your DNA. It's the rules on how to read your DNA. So your DNA must be read in threes, and your mRNAs are also read in threes. So let's talk a little bit more about the genetic code by looking at the codon and anticodon. The codon is genetic information in mRNA that exists in triplet bases. So we're talking about 3 nucleotides at one time. A codon can code for a specific amino acid, or it can code for the start or stop of transcription or translation. Okay, so if your codon is coding for specific amino acids, say for example, your codon UGG codes for amino acid tryptophan, shortened to TRP, then you're basically taking a codon, which is 3 bases in mRNA, looking at that code and understanding that it translates to 1 amino acid. Whereas if your codon is coding for a start or stop codon, that means your codons are signaling the beginning or end of a transcription or translation process. If it is coding for a start codon, first example, then that means your start codon will be AUG, and it begins the translation for that gene. It will also code for amino acid methionine. If your codon is coding as a stop codon, that means it's either UAA, UAG, or UGA. And once the enzymes reach there, it will basically stop the transcription or translation. So this is what the codons and anticodons will look like. So the codons are triplet bases in mRNA. This down here is your mRNA. And this is one codon. As long as you have 3 letters that are being read together, kita baca 3 huruf atau 3 base nucleotide itu sekali, itu adalah satu codon. Your anticodon is the triplet bases in tRNA that is complementary to the codon. So kalau codon adalah di mRNA, anticodon ada di tRNA. Dan dialah yang bermatching dengan codon. So this part in green is an anticodon. Your codon and anticodon will be complementary. That means they have nucleotide bases that match each other. Okay, so let's talk a little bit more about the genetic code. What are some of the features of the genetic code? The genetic code is composed of 4 nucleotides on your mRNA. So it's either adenine, uracil, guanine, or cytosine. And they will be read in triplets. Kita akan baca dia 3/3. So you have 3 nucleotides for 1 codon. There are 64 possible codon combinations. We have the start codon, which is always AUG. It will start the transcription, and also at the same time, it can code for amino acid methionine. And you have the stop codons, which are UAG, UGA, and UAA. So how I try to remember the stop codons is you think of yourself like a caveman. Okay, pretend you are a caveman and you go wah, uga, wah, 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 uga, and then that is basically your stop codons. The code is degenerate or redundant. That means you have more than one codon that can code for the same amino acid but no ambiguity. Apa maksud ayat di sini? Kita punya codon ada yang berulang. Maksudnya beberapa codon boleh code untuk amino acid yang sama. But no ambiguity means that asalkan dia ada 3 huruf itu, dia confirm akan hasilkan amino acid itu. Dia tidak akan, kadang-kadang dia amino acid methionine. Kadang-kadang dia amino acid glycine. No maybes, always this. Okay? No ambiguity. Tiada kekeliruan. And there are non-overlapping. So they don't overlap with each other. Okay, so this is what we are talking about when we say codons matching up to your amino acids. This is a codon table for mRNA. So according to this table, you should be able to see if your codon is UUU, the amino acid that you will get is phenylalanine. If your codon is AGA or AGG, the amino acid that you will get is arginine. If your amino acid is GCU, GCC, GCA, or GCG, then you will get alanine. And as you can see here, if you get AUG, AUG, then your transcription or translation will start because this is a start codon, and it also codes for methionine. If any of your codons read as UAA, UAG, or UGA, WA, WAK, or UGA, then your transcription or translation will immediately stop. Okay, so this is your codon table. Usually it will be provided for you, but it's good to remember a few common examples. Okay, so that is it for the terms and kind of an introduction to the genetic code. Now we're going to go into the protein synthesis itself. So we already know that protein synthesis is composed of 2 stages, which is transcription and translation. We are going to start with the first stage, transcription. Transcription is the process that will occur in the nucleus, and it involves 3 stages, which are initiation, elongation, and termination. It basically involves these 4 things. First, it involves your DNA. Only one strand of your DNA will act as a template. Second, it involves the enzyme RNA polymerase. RNA polymerase will add RNA nucleotides. Third is a promoter, which is a region in your DNA with a start signal. Fourth, a terminator, which is a region in your DNA with a stop signal. So now get ready. We're going to look at the mechanism in each stage of transcription. Beginning, of course, with number 1, initiation. Initiation occurs when RNA polymerase will bind to the promoter and then unwinds DNA strands, separates them into 2 single strands, and form a bubble. So macam mana initiation mula? RNA polymerase akan jumpa promoter, yaitu satu kawasan pada DNA, yang tempat mula kamu punya transcription. So bila dia bind pergi promoter, dia pegang kepada promoter, dia akan mula buka DNA strand. Bila dia buka, kita cakap dia unwinds the DNA strands. So now it will separate into two strands and form a bubble. Kamu boleh nampak sini, bila dia buka bentuk bulat, itulah yang dikatakan transcription bubble. And RNA polymerase initiates mRNA synthesis by adding RNA nucleotides that are complement to the template base, which is DNA. So asal dia buka saja, dia akan mula sudah transcription process. Macam mana dia mula? Asal dia boleh baca, dia terus cari RNA nucleotide yang match, yang complementary, dan dia akan masukkan dan start susun. Only one of your DNA strand will act as a template and is copied. So, kalau macam contoh ini, yang 3' to 5' end, which is the bottom one, will be the template strand. All right, now we can go on to the second stage of transcription, which is elongation. Macam mana mRNA strand kita ini memanjang? Okay, after initiation, the RNA polymerase will move downstream, unwinding the DNA and elongating the RNA transcript from 5' to 3' direction by adding nucleotides to the 3' end of the growing polymer. So that apa maksud ayat di sini adalah RNA polymerase akan bergerak turun, okay, dari kiri ke kanan, downstream. Sambil dia bergerak, dia akan buka DNA dan terus menambah RNA nucleotide untuk memanjangkan mRNA. We can call this growing RNA strand, growing mRNA, elongating RNA transcript, whatever it is, make sure you are pointing out that it is the RNA. Okay, dia akan memanjang dari 5 ke 3, 5' to 3' direction. Behind the point of RNA synthesis, the DNA double helix will reform and mRNA is released. Where the RNA synthesis has passed, okay, RNA polymerase sudah lalu, tiada benda akan pegang DNA dan dia akan terus gabung semula. Gabung semula adalah bahagian ini. DNA double helix reforms. And then the mRNA will be released. Dia akan berjuntai di belakang. Okay, sometimes your cells need to produce large amounts of protein, of the same protein. So it will transcribe the same gene many times. The same gene will be transcribed by several RNA polymerase simultaneously to produce many mRNA. So kalau dia mau hasilkan banyak protein yang sama, dia kasih banyakkan RNA polymerase. Okay, satu sudah lalu, terus masukkan yang kedua. Kedua baru sampai setengah, masukkan yang ketiga. Untuk sel kita hasilkan lebih banyak protein yang sama. Okay, so after elongation, we will reach termination. Termination means ending, okay? Penutupan. Transcription will terminate or stop when the RNA polymerase reaches a stop signal at termination site. Termination site adalah kawasan di mana kamu punya transcription memang sepatutnya berhenti. Dia akan ada satu stop signal. Your mRNA or RNA transcript is then released. The RNA polymerase terbuka, mRNA kamu pun terbuka. And they're both detached from DNA. And that's it. That's how your transcription process will finish. After transcription, you will have RNA processing, but this occurs in eukaryotes only. RNA processing is also called pre-mRNA process. mRNA is modified before moving out into the cytoplasm. Modified means we will add a 5' cap at the 5' end and a poly-A tail at the 3' end. But that's not all we're doing. Kita bukan saja tambah satu penutup di hujung dan satu ekor di hujung. Kita juga akan buat splicing. Our cells will also do some splicing. So what is RNA splicing? It is the removal of introns that are joining the exons. And then joining the exons. Apa benda ni intron dengan exon? Well, your mRNA as a eukaryote contains exons and introns. Exons are the coding sequences that will encode a protein. So it is the useful part of your mRNA. Whereas the intron is the non-coding sequence. Intron, saya suka paham perkataan intron ni sebagai introduction. Biasa kadang-kadang kita buat pendahuluan, kita macam bagi banyak ayat-ayat bunga kan. Okey, jadi kamu anggap saja intron ini sebagai ayat berbunga-bunga, tapi actually tiada isi kandungan yang penting. Okay, exon itu yang ada maklumat penting yang kita punya sel perlukan. Now, RNA splicing and pre-mRNA process is only in eukaryotes because in prokaryotes do not have introns and exons. Eukaryot saja ada intron dengan exon. Itulah sebabnya eukaryot saja yang perlukan RNA processing. Okay, so after RNA splicing, we talk about why it is important for us to do the RNA processing. RNA processing is important to facilitate the export of the mature mRNA. Kalau sudah splice, lebih mudah untuk mRNA itu dipindahkan daripada nukleus kepada cytoplasm. And it is also to protect the mRNA and prevent it from rapid degradation by the hydrolytic enzymes. Sebab kita tambah itu cap dengan itu poly A tail, okay, then kita punya mRNA ini lebih selamat. Enzim-enzim dalam cytoplasm tidak akan terus potong dan hadam kita punya mRNA. And RNA processing also helps the ribosomes attach the 5' end of the mRNA. So dia akan membantu ribosome untuk melekat kepada mRNA kita untuk stage yang seterusnya. Okay, so before we go into the next stage, we also need to talk about translation, what it is, and the RNAs that are involved. So the next stage is protein synthesis translation. It occurs in the cytoplasm, involves 3 stages: initiation, elongation, termination, and involves 3 types of RNA. The 3 types of RNA that are involved in translation are mRNA, rRNA, and tRNA. mRNA, kita sudah cakap dari tadi, itu adalah messenger RNA. And let's talk about rRNA, ataupun kita boleh gelar dia sebagai ribosomal RNA. So this ribosomal RNA, as you can see from this diagram, it consists of 2 subunits. It has a large subunit, bagian yang lebih besar, and a small subunit. Looking at the large subunit has 3 binding sites. If you look at this diagram right here, okay, this top diagram is a computer model of a functioning ribosome. Ini adalah rekaan melalui komputer, okay, computer modeling. But if you are to shrink down to the size of these molecules, to the size of atoms, this is probably how your ribosome will look like. Kalau kamu dapat jadi kecil pada size atom, kemungkinan inilah bentuk ribosome yang kamu akan nampak bila kamu bentukan. Okay, so there are 3 binding sites inside. It's not very clear with the first diagram, but it's much clearer with the schematic model. Schematic model ini adalah lukisan mengikut pemahaman kita. Mengikut pemahaman kita, kita boleh nampak ada 3 site di mana tRNA boleh melekat, yaitu A site, P site, dan E site. The P site, the one in the middle, also called the peptidyl site, it holds the tRNA that is carrying the growing polypeptide chain. So nanti bila ada tRNA masuk, salah satu daripada mereka akan memegang polypeptide chain ataupun protein chain yang sedang memanjang. Dan tRNA yang pegang polypeptide chain yang sedang memanjang itu akan duduk di tengah-tengah, yaitu di P site. The other site is A site, amino acid. This is the site that holds the tRNA carrying the next amino acid. Okay, amino acid yang seterusnya yang mau disambung akan masuk melalui A site. Dan E site is exit. So this is for tRNA that already gives up their amino acid for them to exit from the ribosome. Ini adalah pintu keluar. So that is the large subunit. Let us look at the small subunit. Small subunit, the part under here, the function of the small subunit is to hold tRNA and mRNA close together and catalyzes the addition of amino acid to the carboxyl end of the growing polypeptide chain. So, kerja dia adalah membantu. Membantu kasih dekat tRNA dengan mRNA dan juga membantu penambahan amino acid kepada carboxyl end. Okay, so that is it for our RNA. Let us look at the other RNA involved, that is tRNA or transfer RNA. This is what tRNA looks like. It has what people usually describe as a hairpin shape, bentuk macam pin rambut. tRNA is made in the nucleus using DNA templates, and then when they are ready, they move out into the cytoplasm. The function of tRNA, nama dia transfer RNA kan. Jadi fungsi dia adalah untuk transfer amino acid from the cytosol to ribosome and they can be used repeatedly. Fungsi dia adalah untuk membawa amino acid pas kepada ribosome dan boleh digunakan berkali-kali. Its structure. How is the structure of the tRNA? Your tRNA will have an empty codon, and the 3' end of the strand is the attachment site for amino acid. Apakah yang maksudkan dengan point ini? Okay, if you're looking at this diagram, that little purple bit at the end, okay, yang macam purple sikit di hujung itu, sanalah di mana kita punya amino acid akan melekat. Okay, ada tiga cara untuk kamu lukis tRNA. Cara pertama adalah bentuk hairpin macam ni. So this is the first. This is the first of the 3 ways to draw tRNA. This is the second way to draw tRNA. This is the 3-dimensional structure. And this is my favorite way to draw tRNA because it's the easiest. This is the symbol version. Kalau bentuk simbol ni, kamu tengok dia macam lilin yang bengkok. Okay, yang macam tali lilin itu adalah tempat di mana amino acid akan melekat. Dan di bawah dia ada 3 kaki. 3 kaki itu adalah dia punya anti-codon. So if you ever need to do a drawing of tRNA, I usually recommend the third method because it is easiest. Okay, before we can go on and talk about translation, we need to talk about activation of amino acid for attachment to tRNA. So, macam mana amino acid itu digabungkan atau dilekatkan kepada tRNA? Dia perlukan satu enzim untuk bantu dia. Amino acid is joined to the correct tRNA at the 3' end by an enzyme called amino acyl tRNA synthetase. Amino acid tRNA synthetase. Tukang kasih sambung amino acyl kepada tRNA. The active site of each type of amino acyl tRNA synthetase fits only a specific combination of amino acid and tRNA. So that means enzyme ini, permukaan dia yang di sini, active site dia, hanya sesuai untuk amino acid yang spesifik dan tRNA yang spesifik. Tidak boleh calang-calang tRNA datang duduk di sana. Mesti yang ngam dia punya sequence. So since we have 20 different amino acids, we have 20 different amino acyl tRNA synthetase. 20 enzim untuk 20 amino acid yang berbeza. Okay, so that is kind of the introduction. Now you are ready to learn about the mechanism in translation. We're going to go into the mechanism in the 3 stages of translation, which are initiation, elongation, and termination. If you're ready, let's go! Okay, first stage, initiation of translation. We start with a small ribosomal subunit, which will bind to the mRNA at the binding site. Yang bahagian bawah ini dia akan melekat kepada mRNA pada mRNA binding site. And then the initiator tRNA, which is the first tRNA to start, with the anticodon UAC carrying methionine, will bind to the start codon AUG. Dia akan datang duluan, dia melekat kepada start codon. And then, finally, you will have your large ribosomal subunit bind to form translation initiation complex. So, bawah dulu, kemudian initiator tRNA, kemudian ditutup dengan large ribosomal subunit. Bila large ribosomal subunit sudah datang dan gabung begini, ini digelar sebagai translation initiation complex. Perkumpulan ataupun kompleks untuk memulakan translation. Proteins called initiation factors are required to bring all the translation components together. Ada protein lain yang membantu. Protein lain itu digelar sebagai initiation factors. Okay, dia akan tolong bawa kepada sini. Tapi dalam diagram ini kita tidak tunjukkan secara jelas mana itu initiation factors. Okay, we are going to go into the next stage of translation, which is elongation. This one will be a cycle. So let's look at it section by section. Elongation of translation consists of a series of 3-step cycles as each amino acid is added to the preceding ones. Setiap kali kita tambah satu amino acid, kita ulang satu kitaran. Kitaran ini melibatkan codon recognition, peptide bond formation, and translocation. Let's look at the first substep in elongation, which is codon recognition. This one. Codon recognition mengenal codon. So in the first step, codon recognition dia mengenali codon yang ada di sana. So the anticodon of the incoming tRNA will pair with the complementary mRNA codon on A site. Dia akan tengok apa codon yang ada di A site. tRNA yang akan datang adalah yang matching dengan codon di A site. You also have elongation factors or proteins that assist in hydrogen bonding between the mRNA codon with the anticodon of tRNA carrying the correct amino acid. Apa maksud poin kedua adalah dalam gambar raja ini tidak ditunjukkan, tapi ada protein-protein lain yang membantu tRNA kita untuk bind dengan mRNA. Dia bantu hydrogen bond formation di antara anticodon dengan codon. And then you also have hydrolysis of GTP, guanine triphosphate, to increase the accuracy and efficiency of the steps. Okay, so they're going to be here to help in the codon recognition. The second step in elongation part of translation is the peptide bond formation. So it is happening here. In the peptide bond formation step, rRNA will catalyze the formation of peptide bond between the polypeptide on tRNA in the P site with the new amino acid on tRNA in the A site. Dalam erti kata lain, dia akan membantu untuk hubungkan amino acid yang di sini, yang di P site, dengan amino acid di A site. You will see a little red arrow over there. That is what is happening. That's the peptide bond formation. So in this step, it will separate the tRNA at the P site from the growing peptide chain and transfers the chain to the tRNA at the A site. Kamu boleh tengok gambar raja di sini. Mula-mula polypeptide chain itu berada di tRNA yang di tengah-tengah, yang di P site. Selepas step kedua ini, growing polypeptide chain akan bersambung kepada tRNA yang berada pada A site, yang di tepi. So now the middle one is empty. So that will bring us to the third step in elongation of translation, which is translocation. Okay, it occurs here. This is where your ribosome will translocate the tRNA with the polypeptide chain from A site to P site. Translocation juga boleh diringkaskan kepada konsep perpindahan. So sekarang kita akan pindah, ribosom ni berpindah. Bila dia berpindah, yang tadi di A akan duduk di P site. Yang tadi di P site pula akan pergi kepada E site. So the empty tRNA that was previously in the P site is now moved to the E site and then released. Because of the tRNA anticodon that remains bonded to the mRNA codon, the mRNA will move along with it. So mRNA pun akan ikut bergerak bersama. So now the next codon is available at the A site. Sekarang A site kosong lagi, codon seterusnya ready untuk next tRNA. Translocation ensures that mRNA is read from 5' to 3' direction, codon by codon. Sebab ribosom ini akan bergerak 3 nukleotide demi 3 nukleotide, dia akan pastikan kita punya mRNA sentiasa dibaca dalam arah yang betul dan sentiasa dibaca codon demi codon, 3 by 3. Okay, so now we just need to terminate the translation. This translation will continue until it reaches a termination codon or a stop codon. So elongation continues until it reaches stop codon, until the stop codon reaches the A site. And then you will get something called a release factor. The release factor will bind to the stop codon and hydrolyze the bond between polypeptide and tRNA in the P site. Maksud ayat kedua ini adalah bila release factor sampai di A site, release factor itu akan berngam dengan stop codon. Jadi di bawah dia boleh berngam dengan UAG, UAA atau UGA. Dia sampai sudah di sini, dia akan putuskan bond di antara polypeptide chain dan tRNA. It's basically cutting the connection between the protein and the tRNA. After that, this frees the polypeptide, and the translation complex will disassemble in a multi-step process. Selepas dia potong tu, polypeptide tu bebas, terapung-apung dalam cytoplasm, dan kemudian translation complex ini akan dibuka satu demi satu. Okay, we are not learning the disassembly process. We only learn the translation itself. Okay. Another thing that we are learning about is the concept of polyribosomes or polysomes. Kadang-kadang memang satu ribosom sahaja untuk satu mRNA, tapi bila kita punya sel atau kita punya badan perlukan banyak protein yang sama, daripada dia balik-balik buat banyak salinan mRNA, apa dia boleh buat adalah dia guna satu mRNA dan ambil banyak ribosom. Polyribosomes is when you have several ribosomes on one single mRNA simultaneously. So what happens is it can produce identical polypeptides in large amounts in a short time. Sebab diorang semua sedang translate pada masa yang sama. Satu mRNA boleh digunakan untuk menghasilkan salinan yang banyak. You can use one mRNA to make many copies of a polypeptide simultaneously. The polypeptides will undergo modification before being used as a particular protein. Jadi selepas translation ni, kita punya polypeptide tu yang dibebaskan akan ada dia punya pemprosesan lagi di organel lain sebelum dia ready untuk digunakan. Okay, so that is it for polyribosomes. Okay, so that is it's very short, but take your time to read and understand and really see if you can remember the steps that are involved. Just a quick summary about what we learned in 6.3 is that you have the genetic code, usually referring to your codons. The codons will code for amino acids. When we are talking about protein synthesis, it usually involve not usually, it always involves transcription and translation. So transcription will have initiation, elongation, and termination. Translation will also have initiation, elongation, and termination as well. There is also another video lesson for protein synthesis.
In
Unfortunately,
order
this
for our
video is
bodies
no
to
longer
function,
available on
we need
YouTube,
to supply them
but
with a
we do
variety of
have a
nutrients we get
copy
from our
of
diet.
this video
Our
in
bodies cannot
your
use the food
lecture
as it
notes. So
is
if
when
you're
it
using
enters our
this lecture
digestive
note
system.
in
The
PDF
process of
form, you will
chemical
not be able to
digestion
play the
uses
video,
different
but if you're
proteins and
looking at it in
enzymes to
PowerPoint
break down
form,
the food
you can
particles
get
into usable
the video to
nutrients
play.
our cells can
I
absorb.
may also send
And where
this
are the
video
instructions
in
to
the Telegram
manufacture
groups
these and all the
if
different
you're
types of
really
proteins we need
unable
to
to
stay
use it
alive?
in PowerPoint.
The instructions to make
Okay, so do have
proteins
a
are
look. It
contained
has
in
a
our
really good
DNA.
explanation. And
DNA
I
contains
really hope that it
genes.
helps
A
you to
gene is a
visualize
continuous
how
string of
protein
nucleotides
synthesis
containing a
involving
region
both
that codes for an
transcription
RNA
and
molecule.
translation
This
occurs
region
in each
begins
of
with a
our cells.
promoter and ends
Okay.
in order.
So that is it for 6.3. What should you do now? Well, you should make your own notes for 6.3 and answer the Google form quiz for this subtopic, which can be found in your Google Classroom. That is pretty much it for me for this lesson. Thank you very much for your time and attention, and I will see you again soon. Happy learning and goodbye.